Good afternoon. Our speaker today here at CEDAD is Rizwan AZIZ, a young postdoctoral researcher who is making training on electron holography at the Bio Open Lab at the Department of Mathematics and Physics at the University of Salento. The Bio Open Lab is in fact based on a 200 kilovolt atomic resolution holo-TEM manufactured by GEOL in Japan. The facility is already an associated facility of the European consortium CERIC-ERIC and is open to researchers from all over the world. The deadline for the next call will be in September. Rizwan AZIZ received a master's degree in physics at the National University of Science and Technology in Islamabad, Pakistan. And he received a PhD in physics and nanoscience at the University of Salento. Now he's a postdoctoral research scientist at the University of Salento. His research is about functional tests and developing new methods of electron holography on various kinds of materials. In the seminar, he will trace the historical roots and milestones of electron holography, and he will explain the differences and advantages of inline and offline electron holography. He will show how electron holography can form, record, and reconstruct interference patterns that reveal the morphology, phase, electron potentials of the samples. He will describe some applications of electron holography in biology, such as visualizing protein structures, cellular membranes, and molecular interactions. The title of the seminar is Electron Holography, Principle, Theory, and Biological Applications. The stage is yours.
Buon pomeriggio a tutti. Grazie professore Lucio Calcanile per avermi dato questa opportunità. Il titolo della mia presentazione è Olografia elettronica, principi, teoria e applicazione biologiche. This talk actually tries to encompass the idea of electron holography, a technique that uses the interference pattern of electron beam to reconstruct the image and phase properties of a specimen. I will start with the introduction of electron holography, then I will give a brief historical perspective of the technique. Then I will throw a little light on principle and types of electron holography with the emphasis on basis of off-axis electron holography. Then I will conclude with some biological applications, some future prospects of this technique. Here is the schematic of the microscope GeoGem Arm 200F, NEO arm, which is installed at the Department of Physics and Mathematics at University of Salento as part of the BioOpenLab project, presided by Professor Lucio Calcanile. The microscope, we can see that it has the basic components that like cold field emission electron gun, which is obviously extremely advanced to ensure the coherent beam of electrons. Then there is a series of condenser and objective lens and apertures, and one of the most important thing is the aberration correctors. It has a state-of-the-art aberration corrector that can limit the aberration up to 5th order. And then the most indispensable part is the electron biprism that actually ensures the electron holography technique to be realized. I will explain in little more detail in the upcoming slides. So I will begin with a little historical perspective. In 1897, very quickly and briefly, J.J. Thomson detected the electron as first elementary particle, and then Louis T. Broglie said that in his researches on the theory of quanta research paper, he said that each particle there is a wavelength associated that can be calculated with this formula. Then the first diffraction experiment was performed using the electrons and the nickel surface, and it was performed by the Davison and Germer in 1927. And then Dirac in 1933 postulated the single particle interference, which is again very important because Dirac single particle interference tells us that we don't need multiple particles for the interference to be generated. Just the single particle can interfere with itself. So in 1934, the first transmission electron microscope was invented by Ruska, and in 1936, Bosch introduced the diffraction lens in the TEM, and which is obviously to show that the electron microscope is a powerful diffractor. Kosel and Molenst developed conversion beam electron diffraction in 1939, and afterwards in 1940, Bosch found Fresnel fringes as a diffraction phenomena at edges in the transmission electron microscope. And then this is the most important part, where in 1948, Dennis Geber introduced the concept of holography in his famous paper, A New Microscopic Principle. And on this side, we can see, we can see that the Dennis Geber originally introduced the idea of holography in the form of inline electron holography. Then in the first electron hologram was recorded in this experiment by Hain and Mulby in 1952. The formation of the diffraction image with electrons in the Geber diffraction microscope. So the microscope idea was built by Geber, and under his supervision, Mulvey and Hayne constructed the first hologram with a microscope of 60 kilo electron volt, and they were able to achieve a resolution of 10 angstrom. We can see the image of the first electron biprism. That is extremely, as I said before, important part to generate the interference pattern. And it was originally developed by the Molenst and Duker. Duker was his PhD student, so together they were working on an experiment. And the results of the very first experiments are shown here, where by increasing the voltage, we can see that the interference pattern is more wider, and there are more fringes inside the hologram. And why? Because the voltage of the biprism affects the interference of the two parts of the electron beam. We will see in more detail in the upcoming slide with the mathematical relationship. So what is electron holography? Electron holography is a technique that uses the wave nature of electrons to construct an interference pattern and to record and reconstruct their image and phase information of the specimen under observation. So the phase information, why the phase information is important? Because the phase information can reveal both the electrostatic and magnetic field information inside the specimen. And there are many kinds of holography techniques developed over the years, but I will try to just focus myself on two of these, off-axis and inline electron holography. Otherwise, there is also a point projection and other schemes suggested afterwards. Okay, so the two main types of the holography that I'm going to be focusing on is inline electron holography and off-axis electron holography. So here is the schematic of the inline electron holography. In inline electron holography is a method of imaging objects by recording and reconstructing the phase and amplitude information. And we can see that in inline electron holography, both object and image are being formed in the same optical plane. For instance, the central part, which is dark blue, is the electron part of the electron beam that has passed through the specimen, and the periphery part of the electron beam actually interferes with the central part, and together they form an interference pattern on the detector. At that time, there were some limitations of adopting this technique. What were those limitations? First of all, the spatial and temporal coherence of the electron beam was not wide or long enough to realize this kind of technique. And secondly, the incoherent scattering effects added an unwanted strong background. As I said before, that because the image and its conjugate image are formed in the same optical plane, so with this technique we come across a very significant problem of twin imaging. So because of the geometrical limitations of the inline electron holography, the off-axis electron holography was invented. And in this kind of holography, we use an electron biprism to split and recombine an electron beam into an object wave and a reference wave, and which forms an interference pattern, which is known as the hologram on the detector. And this hologram actually encodes the information of both image and phase of the specimen. Here is a little simulation of how off-axis electron holography works. These are the components: cold field emission electron gun. There is a specimen, objective lens. I'm denoting all the stigmators and other lenses with just one for the sake of simplicity. And there is an electron biprism. So we can see that from the cold field emission electron gun, there is a beam of electron, half of which passes through the specimen, half of which passes through the vacuum. And then in the back focal plane, this electron biprism, because of its positive voltage, it converges the two parts of the beam and forms a hologram, an interference pattern. What are the bases of off-axis electron holography? I have divided them into three parts. First of all, I will try to explain what are the factors that are involved in the hologram formation, what are the factors, how we record, And the third most important part is the reconstruction of the hologram. So first of all, the coherent electron sources. I will try to shed light why the coherence of the electron beam is important to realize the electron holography. Here is a bird's eye view of the electron gun systems, thermionic emission guns and the field emission guns. And there is a comparison between these two. One of the most important factors is the energy spread of the electron beam, because the cold field emission electron gun has the least energy spread, which means that it will suffer less chromatic aberrations and other kinds of aberrations. Although it's extremely expensive and very delicate, but there are many advantages that ensure that the cold field emission electron gun actually generates a coherent beam. So this gives rise to two concepts that need explanation. What is the temporal coherence and the spatial coherence of the electron beam? So the effect of the finite size of the electron gun actually determines the spatial coherence of the electron beam, and then the temporal coherence is determined by the energy spread of the electron beam. So that's why I was saying that why the energy spread, lesser energy spread is so important to ensure the temporal coherence of the electron beam. And both of these spatial and temporal coherences can be determined by these formula. So here is a schematic representation of, just to understand that we can see a schematic diagram in which one plane wave is propagating from left to right, and there are two other plane waves that are propagating at plus beta and negative beta angles with respect to this plane. So we can see that in the vertical direction, these plane waves actually converge together, they mix together, and they lose coherence. So up to this length, Ls is equal to lambda over 2 beta, these electron beams actually remain coherent. So they can be definable. So this length up to which the electron beams at different angles remain coherent is called the spatial coherence length of the electron beam. And again, the temporal coherence can be, in the same way, can be described that up from the certain length in time, the electron beam will lose its coherence. So these are the concepts of spatial and temporal coherence. The second part is the electron biprism. It was the device that was built first, as I explained before, by the Molenst and Duker in 1953. And it actually enables the two parts of the electron beam to converge together to form electron hologram. So the electron biprism installed in the transmission electron microscope, Jewel GEM-ARM 200F, these are the characteristics of that electron biprism, that it is 0.6 micrometer diameter, and it has the length of 1.7 millimeter. And in the later slides, I will show with the certain mathematical relationship that how the width of this electron biprism actually affects the final hologram formation, and how the width of the electron biprism actually determines the spatial resolution of the image and the face. So one of the simplest way to understand the action of the electron biprism is shown here, where the deflection angle alpha is actually directly proportional to the applied voltage on the electron biprism. So if the applied voltage on the electron biprism is higher, then these two parts of the electron beam will converge more excessively together, and they will form a wider hologram on the detector screen. So where Kf is the deflection coefficient and its value is 10 to the power minus 6 radians per volt, and it depends upon the thickness of the electron biprism. And we can see that there is a linear relationship between the angle of deflection and the voltage applied on the electron biprism. So it means that if we need a hologram of wider width, we can simply increase the voltage of the electron biprism, but again up to a certain limit. So from these formulas, it is even easier to understand. Here we can see a schematic of the action of the electron biprism in which the two parts of the beam can be considered as the two different sources. Here the interference width in the hologram is given by this formula, where alpha is the deflection angle and r is the radius of the electron biprism. The fringe spacing S is given by this formula. We have to note that the width of the electron biprism is directly proportional to the deflection angle, and spacing is inversely proportional to the deflection angle. So if the deflection angle is higher, the width of the hologram will be more and the interference fringe spacing will be much less. And this is what we actually want to obtain a highly resolving image and phase information from the specimen. So here is the potential on the biprism, the relationship of potential on the biprism. Here we have the phase shift suffered by the electron wave and the angular deflection. So angular deflection is directly dependent on the line charge density. So line charge density through this equation is related with the potential. And the potential, it means that by increasing the charge on the electron biprism, potential increases and then the width increases, width of the hologram. Another important factor that we can explain from this equation is the width of the electron biprism. So we can see that the width of the electron biprism casts a shadow that can be seen in the image if the voltage is 0. So we need to keep the width as minimum as possible to avoid the formation of the Fresnel diffraction around the image of the transmission electron microscopy image. So the last part of the last factor that is involved in the holograph formation is the aberration correction system. So first of all, we need to understand what are aberrations. There are actually three kinds of aberration: geometrical aberrations, chromatic aberrations, and parasitic aberrations. So I will try to focus just on the two of the most commonly occurring aberrations, chromatic and spherical aberration. So what is a spherical aberration? It is caused by the deviation of the, we can see here, of the electron rays from an ideal spherical wavefront due to the imperfections of the magnetic lenses. So electrons passing through the periphery of the lens are refracted more than the electrons that are passing through the center of the lens, which means that they don't reach a common focal point, so that causes the blur in the final image. So here is the relationship of the aberration coefficient, spherical aberration coefficient, and one way to limit this kind of spherical aberration is to increase the excitation voltage or excitation current along the objective lens, but again only up to the optimal value, because if we increase the excitation current along the objective lens beyond an optimal point, then it gives rise to even more aberrations. So we need to understand what is the optimal value of that current in which we could have the minimum value of CS, spherical aberration corrector. So here we have the chromatic aberration. So what is chromatic aberration? We can see in these videos. First we can see the spherical aberration and then the chromatic aberration. The difference in the colors. In the case of electron beam, it shows that the electron have different energies. So when they have different energies, they will have different wavelengths. So different wavelengths of the electron beam will be converged at different points from the lens, and this gives rise to the chromatic aberration. But again, there are several factors that determine the chromatic aberration inside the microscope, one of which is the current that we used in the objective lens. So we can see the one factor is the excitation current, the other factor is the accelerating voltage, and then the energy spread. You see, again, the energy spread is affecting the aberrations. And the last is the inelastic scattering of the electrons as it passes through the specimen. An effective way to reduce the CS, the spherical aberration inside the microscope, is to to use a aberration corrector device, and which is a device that consists of several magnetic lenses and deflectors that can compensate for the deviation of the electron rays caused by the aberration. So one of the most specialized devices that this microscope has is a pair of the aberration correctors, both of which are known as SESCOR corrector, and it can correct the aberrations up to 5th order, and this CS corrector also permits a high flux in the electron probe. So without losing the flux density of the electron, it can compensate for the aberrations up to 5th order, and this leads to a 10 times higher probe current and at the same time to twice the smaller probe diameter on the sample. So there are examples that have shown that by using the same kind of microscope with and without the CS correctors, without CS correctors it has produced the blurry images. So the second part or the third part I will explain together is the recording and reconstruction of the hologram, which is actually the backbone of this process. So the recording and reconstruction can be done by several methods, one of which is the Fourier transform method. And the softwares that are used for this method there are several softwares, like PyHolo is a standalone software. There are actually plugins for digital micrograph. There is HoloWorks, there is Holography. So these softwares actually automatically put an algorithm to reconstruct the electron hologram, but most researchers don't actually prefer to automatically put an algorithm. They develop their own algorithms depending on the initial conditions of their specimen. So we will see in more detail in the research examples. In order to understand the Fourier transform method, it is extremely important to understand what is Fourier transform. So it is a method that transforms a function of certain variables into a function of the variables conjugate to the certain variable. For example, Fourier transform of the sound signal or the light signal, which is a function of the time, can be used to convert it into a function of wave number or frequency. And we can see that in electron microscopy, a Fourier transform can be used to analyze the images and diffraction patterns of the samples. This is the significance of the Fourier transform. So Fourier actually showed that any signal, any periodic signal can be expressed as the sum of the sine waves with various amplitude, frequencies, and phases. So any periodic signal can be expressed in this form. Here we can see that it was a function of the time, and it is Fourier transformed into the function of frequency. Here we can see, as explained earlier, Fourier transform is based on the idea that any image or signal can be decomposed into a sum of sinusoidal waves with different frequencies, amplitude, and phases. So here we can see a square wave, which is a non-sinusoidal wave. So a non-sinusoidal wave is being broken down or is being changed or Fourier transformed into the odd sine functions. Why odd sine function? Because a non-sinusoidal square wave is an odd function, so which is symmetric around the origin. So it can be expressed as the sum of odd functions. So here again, the interpretation of how this non-sinusoidal square wave is composed of different sines functions in the frequency domain. Here is the actual example. This figure shows the Fourier transform of a crystal, gold crystal, and this Fourier transform, then we take an inverse Fourier transform to create the image, or this image and the Fourier transform of the image B. So in microscopy, these are the relationships where the Fourier transform of the scatter waves originating from the object or crystal potential, and then we take the inverse Fourier transform to produce the object image. And in electron holography, not only image but also the phase information. So here we can see there are two functions, psi r and psi k. Psi r is the real space function, which stands for the scattered waves, and this is the function described or Fourier transformed in the reciprocal space. and in the frequency domain. So when we take the inverse, this is inverse Fourier transform. So inverse Fourier transform generates the image as shown in this example. Okay, so now we can move to the last part of the basis of AZIZ electron microscopy, the reconstruction part. The reconstruction, this intensity of the hologram is formed by taking the square of the two wave functions of the two separate parts of the electron wave. And we can see that when we take the Fourier transform of this wave, this intensity of the hologram, we are left with three very important things. There is a central band, there are two side bands. And now we have to decide that which band is needed to be selected and which band actually contains the information of both image and phase. So the central band is actually the conventional image of the specimen, and one of the center band, they are the conjugate bands. So we, in the next slide, let me show you, this is the process of how to reconstruct from this Fourier transform, how to reconstruct the phase and amplitude information from this, this equation. So the center band is, as I explained before, is the Fourier transform of a conventional image. The two side bands are the Fourier transform of the complex image, which contains information of the phase. And what is the process? The process is simple, in which we just cut out one of the side bands. We choose one of the side bands, and then after filtering, we take the inverse Fourier transform of that side band, and that contains both the amplitude and the phase information. And this is the actual example. This is the hologram of AZIZ hologram of the crystal, and then this is how the Fourier transform looks like. There is a central band, there are two conjugate bands, side bands, and one of the side bands is selected, and inverse Fourier transform is taken to generate the phase information. Okay, the final part of the presentation is the biological applications. In the native environment, the biological samples are weak phase materials because they mainly consist of the very lighter elements. So they don't generate enough contrast to be measured through a conventional electron microscopy. And that's why we use these staining techniques, different staining techniques. One of the problems with the staining techniques is that with the staining, it produces many artifacts in the final imaging process. So electron holography is a technique that can image these biological samples without staining. And what is the significance of understanding the electrostatic and magnetic field information from the specimen is because they have many medical applications. They can be used to treat vascular and endocrine disorders, different diseases. Electromagnetic fields can influence also the structure of the proteins. And we know that the protein function actually depends upon their conformations. So if the applied, or if a specimen has a different has a certain amount of electromagnetic fields, it is important to understand that which factor is actually influencing the different conformations of the protein. So electron holography can measure both the electromagnetic fields and the charge distribution in biological systems, also in liquids. So this is again one of the most significant part that cannot be done easily with the conventional transmission electron microscopy. The first example, it's a work from 2017, and it shows the hydrated cell. So this example is extremely important because the researchers were able to construct the image and phase information in the liquid environment for the Magnetospirillum magneticum bacterium strain, AMB1, and assemblies of magnetic nanoparticles using electron holography and off-axis electron holography. So this figure shows an off-axis electron hologram of a bacterial specimen in the liquid cell, and this is the reconstructed wrapped phase image and which is mainly dominated by the mean inner potential, which corresponds to the thickness of this bacterium. And this is the reconstructed image information, and we can see that it still shows the bacterial cell in its original form without any changes. So this set of pictures show that, okay, this is again the off-axis hologram of the bacterium in the initial state, and after tilting by 75 degrees and then applying a magnetic field in the electron beam direction. Why the magnetic field? Because this is actually done in the Lorentzian mode. So then again after tilting and applying the magnetic field at minus 75 degrees. Why it is being done? This technique is called the magnetization reversal. So this technique, the first in the positive 75 degrees, then in the negative 75 degrees. So through the magnetization reversal, we can understand and extract the information, the magnetic field information from the specimen under consideration. So here there is, what are these things in the yellow and red outlines is the, it shows the progression of voids, and it is because of the constant exposure of electron beams, and that's how the electron beams are changing the structure of the liquid, not the native structure of the bacterium. And with the exposure time, this structure is being evolved more. Here we can see the magnetic induction map of the magnetite chain, and the outline of the individual magnetite nanocrystals are marked in white, and these arrows and the color actually show the direction of the magnetic field through the help of this color wheel. Here we have the quantitative representation of the magnetic field that is measured across this dashed red line. Like this magnetic field induction is not extracted from the whole bacterium, but it is only measured across the one cell of the bacterium along this dashed line. We can see that this part is not resolved by the hologram, which is because of the bubbling effect of the incident electron beams. And here, which causes the heating effect in the liquid, and that's why it was not resolvable completely. So the phase profile is used to estimate the in-plane magnetic field induction across the particle. And how to achieve it? The researchers, they treated the particles as a sphere. They are not perfect sphere, but we have to move on with certain kinds of constraints. And so this was like the researchers set the particles as spheres, and then the maximum and minimum values of the magnetic contributions were calculated, and the difference was computed and put in this equation, which helped to calculate the quantitative measurement of the magnetic field induction. Another example, this is actually again one of the most important, because this is one of the first examples in which the researchers were able to construct the structure of a single protein. So extremely important again. So why it is important? This is crucial because proteins are flexible objects that can assume distinct conformations and often associated with the different functions. So if they have different conformations, they will give rise to different functions. So it is extremely important to be able to map the single protein structure to see that, to develop the drugs for that protein, and to have more knowledge of the structure. And here is again the inline electron holography technique is being used. On the graphene, the protein was deposited, and they use these are actually the hologram reconstruction of the cytochrome C molecule. And we can see that it not only generates the information of the imaging information of the single protein, but also it gives the information when it has different orientations and when it has it is forming different agglomerates. So we can see here is the single protein, and then it forms two agglomerates, and it forms three agglomerates. So even like it gives very deep details of the single molecular structure. Here we can see the comparison between the low energy electron micrograph of another protein, BSA, which is a little bit larger than the cytochrome C protein. So, we can see there is a comparison between the model developed by the protein data bank, and we can see that they are in extremely perfectly in agreement with each other. So, these are the holograms of BSA proteins in different orientations, and this is the data based on the protein data bank. So in different orientation, we can see that how deeply and correctly the electron holography can map the original structures. And another way to analyze the micrographs is to compare them with simulated electron density maps. So here we can see that electron density map again are in perfect agreement with the holograms generated by the inline electron holography. Another application of the electron holography in the same example is that it can be used to study the diffusion of the protein. So these are the holograms that were generated after 30 seconds each. So some of the proteins actually moved across. So this technique can also be used to study what are the surface properties of certain proteins, surface diffusibility. Okay, another example, this is actually much more improved because more recent in 2022. Again, they use the inline electron holography and certain algorithms, the algorithms that I was talking about earlier. We will see that which kind of algorithms did they use. And these are the iterative amplitude and phase information of the single proteins. So with the low energy electron holography, it is possible to retrieve the amplitude and phase distribution from experimentally acquired low energy electron holograms. So from the single iteration, we can generate the amplitude information, but to get the phase information, the researchers had to apply several iterations to generate this. We can see the schematic of the hologram generation and the algorithm that was applied. So here is the schematic. We can see that the emitter is producing the electron beam, which passes through the object, and then it creates the hologram on the detector screen. And here is the schematic representation of the iterative reconstruction algorithm. We can see that here is the hologram plane, and by taking the inverse Fourier transform, it comes to the object plane. Another important thing in this example is the concept of constraints, and I will explain in the later slide what does that mean in the formation of the hologram. But for now, we can see that after no iterations, this is the information of the single protein, and after 5, 10, 15, 25. And why they stopped at 25 iterations? Because the algorithm was converging at the 25th iteration, so they didn't have to go further. So here we can clearly see how the image and both the phase of the single protein is improving with the iterations. Okay, now comes the concept of the constraints we saw in the previous slides. It shows without phase constraint, and it shows with phase constraint, and without phase constraint, with phase constraint. So we can see that there is a clear improvement in the hologram, both the image and the phase information. So phase constraint and amplitude constraint are the constraints that the researchers can put before developing an algorithm. So they can be based on the initial conditions, they can be based on the information from the specimen, or they can be based on the general perspective. For instance, one of the amplitude constraint could be that the energy will always conserve during the microscopy process. Or the other amplitude constraint could be that if the electron beam goes through the specimen, it will always lose the intensity, like the intensity will never increase. So we can put a constraint that the intensity of the incoming electron beam would always be less than or equal to 1. So this could be another constraint on the amplitude. On the other hand, the phase constraint could be that all the places where the phase exceeds the maximum value 1, then the algorithm automatically puts it to 0. What does that mean? That means that the phase contributed by the twin image will be eliminated completely. So we can see that how important it is that the twin image problem that I was discussing in the very first slides that are associated with the inline electron holography, the researchers are able to cope with this problem by putting constraints. So when they put this kind of constraint on the phase, they can avoid the contribution from the twin images, and it can be very clearly seen in the holograms. Another example shows the suppression of the twin image. So these actually holograms are being generated by putting both amplitude and phase constraints. So after putting both constraints, we can see there is a clear improvement. There are no, these fringes actually are being generated by the twin imaging problem. So after putting these amplitude and phase constraint, we can see that these fringes are getting disappeared. So, and also the image resolution has improved, and the phase information is even more easily calculatable. The last example again is the mapping of the, this paper was published in 2023, so it's extremely recent, where we can see that two different conformations of the transferrin molecule. So APO and HOLO. APO is represented by the addition of the iron ions, and HALO is the opposite conformation form. And these are the one-step reconstruction of the hologram of both forms. So this appearance of the cleft lobe that was that the researchers were able to see through the electron holography technique actually tells that this is the APO conformation of the transferrin molecule. So even that deeply, the electron holography helps us to understand these different conformations of the same protein. So why the conformations are important, as I described before, that they play a very key role in the biological function of many proteins, and thereby sustaining a multitude of processes essential to life. So when another important thing is that the transferrin molecule is not only attaches with the iron, but also with other elements. So we can also see with the electron holography that which other elements it has attached to, and then what kind of conformation it has achieved. So to conclude, as shown from the different examples, we had seen that the electron holography can reveal the electromagnetic fields, charge distribution, atomic density of the sample with high resolution and sensitivity. It can be applied to study biological samples, even in the liquid form, such as protein. It is able to map the single proteins, cells, tissues, and reveal their structure and function at the molecular level. Electron holography has overcome the phase blindness of the microscopy, and this has helped us to reveal the properties of the nanoscale materials, the magnetic fields, the functional potential in the semiconductors, the strain in the crystals, species of atoms, charge distribution, Coulomb fields around single atoms. So that's it. Grazie mille. Thanks a lot.